Here's an update on our experiment from last week. As you might remember, we planted 100 seeds in 20 different tubes with 5 seeds per tube. Half of the tubes were given gibberellic acid, a plant hormone that causes rapid growth, and half were given water only as a control group. The gibberellic acid group sprouted really quickly and looked great for a few days, but then they all wilted and died. These are all the different parts of a leaf, and this is what a leaf cell looks like up close. Now let's look at the leaf from our plant that was only given water. The leaves from both plants are starting to rehydrate and they look much better. The leaf from the plant that only received water looks totally normal. Now let's look at the leaf from the gibberellic acid plant. Isn't that crazy? All the cells look normal now. The only difference is that this leaf has tiny air pockets all over it. We think these air pockets are full of carbon dioxide, which would explain why the leaf turned black and crispy. It looks pretty normal. But what about the plant that was given gibberellic acid? Whoa! Look how different this leaf looks. But if it's taking in too much carbon dioxide, then it can suffocate and die. If the leaves turn green and the soda lime turns red, then it'll prove that carbon dioxide was a problem. Let's check it out. The leaves look way better and the soda lime has turned red, which means it absorbed some carbon dioxide. This confirms that the plants suffocated due to an excess amount of carbon dioxide. So it seems like the gibberellic acid caused the plants to grow so fast that they took in too much carbon dioxide and suffocated. Anthocyanin is normally purple or red, but it can also appear black in certain circumstances. All the cells are broken down and there's this black, tar-like substance filling the leaf. It looks like the gibberellic acid destroyed the plant's own cells. The control group sprouted more slowly, but they're alive and growing well. Just like anything, too much of a good thing can be a bad thing. So why did the gibberellic acid kill the plants? Today we're going to do another experiment to find out. First, let's check out the roots of the dead plants. So let's rehydrate them and see if that makes a difference. We'll place the leaves in a beaker of water and place the beaker on a heat mat. This will give the leaves the warmth and hydration they need to perk back up. If this does anything, it'll give us a clue as to what killed the plants. They look okay, but let's get up close. The roots appear to be totally fine, which rules out root rot as a cause of death. Next, let's look at the leaves. They seem dry and crispy, but that could just be because they've been out of the water for a few days. While we wait, let's take a look at what a leaf normally looks like under the microscope. But why would this happen? Gibberellic acid is normally used by plants to break down their own cell walls so they can grow faster. It seems like the plants given gibberellic acid grew so fast that they outgrew their own support structure and collapsed. But wouldn't the leaves turn yellow first? What caused them to turn black and crispy instead? Well, when the leaves are submerged in water, we'll start to get some answers. When a leaf is making food through photosynthesis, it takes in carbon dioxide and releases oxygen. To test this, we'll put some spinach leaves in a sealed container with a soda lime, which absorbs carbon dioxide. But why did the leaves turn black instead of yellow? That's because of the anthocyanin pigment, which is made when the plant is stressed. So what's the moral of the story? Well, even though gibberellic acid can help plants grow faster, it can also kill them if they grow too fast. Thanks for watching till the end, and I'll see you next time.